Hello everyone and welcome to this video on nervous system embryology. To truly understand the development of the nervous system, I think it's wise to take a step back to basic embryology. So by the third week of development, we have something called the trilaminar disc. This disc contains three layers: the ectoderm, the mesoderm, and the endoderm. The central nervous system is going to be derived from the ectoderm, and it involves a very important structure called the notochord, which is in brown. This sits in between the mesoderm. This notochord is going to release growth factors to the overlying ectoderm, and these growth factors are going to cause proliferation of ectodermal cells to form a structure called the neural plate. This structure in dark blue is the neural plate, and this is what's going to give us the central nervous system. As the neural plate develops, it's going to go downward and cause the formation of something called neural folds and the neural groove. So as the neural plate moves downward, it's going to form a groove in the center. This is the neural groove, and these sides are called neural folds. We can see a structure in light green. Eventually, when these neural folds converge and fuse, these are going to separate and form neural crest cells. When the neural folds finally fuse, they form this structure called the neural tube. We can see here the neural crest cells overlying them and the ectoderm overlying the neural crest cells. Regarding the development of the brain and the spinal cord, we can divide the neural tube into a cranial and a caudal end. This cranial end is going to give us the brain, and this caudal end is going to give us the spinal cord. If we zoom into this portion, we can talk about the development of the brain. This is a zoomed up picture on the cranial portion of the neural tube. And to develop the brain, the first thing that happens is the formation of primary brain vesicles. These are the primary brain vesicles. The red is the prosencephalon, the blue is the mesencephalon, and the green is the rhombencephalon. And then these primary brain vesicles form secondary brain vesicles. The prosencephalon forms the telencephalon and the diencephalon. The mesencephalon stays as the mesencephalon, and the rhombencephalon forms the metencephalon and the myelencephalon. And an easy way to remember this is that you can think of the telencephalon because it starts with a T at the top, and then everything else is arranged in alphabetical order. The secondary brain vesicles are eventually going to give rise to the structures of the brain. It's really high yield to know what each vesicle derives. I'm going to try to make it easier to remember. However, if you have a basic understanding of neuroanatomy, it's just going to make more sense. So the lowest three vesicles are going to give us the three parts of the brainstem. The myelencephalon is going to give us the medulla. The metencephalon is going to give us the pons, in addition to the cerebellum. The mesencephalon, because it's in the middle of the five vesicles, is going to give us the midbrain. The telencephalon is at the top, so it's going to give the most superficial portion of the brain, which are the cerebral hemispheres and parts of the basal ganglia. The diencephalon is going to give us the thalamus, hypothalamus, retina, and pineal gland. So the walls of these vesicles are going to derive these structures. However, the cavities are going to derive the ventricular system. And it's the same. If you have a basic understanding of the anatomy of the ventricular system, it's going to be much easier to remember these. The telencephalon is going to give the lateral ventricles. The diencephalon is going to give the third ventricle. The mesencephalon is going to give the cerebral aqueduct. The metencephalon is going to give the upper part of the fourth ventricle. And the myelencephalon is going to give the lower part of the fourth ventricle.